moving to the next round
Sige, iwan na yan. Hello, hello.
Hello.
Aldo Guevara Adames. Guevara Adames. Adames. Hello, everyone. Hello.
Hello, ma'am. Hello po.
Ma'am.
Hello. Can I check the attendance now? Can I check the attendance now so that we can start?
Okay po, ma'am.
Okay. Just say present once your name is called. We'll start from section Mango. Epita, Zion,
Presento.
Mark,
Present.
Present.
Mark, Chad,
Present.
John David, Pantiles, John David, Justine, Bihar, Estrobo, Lance,
Present.
Estrobo,
Present.
Go, Hardillo, Malinaw,
Present.
Marasigan,
Present.
Nicart,
Present.
Ogale, Panis, Seso, Sulayaw,
Present.
Uy, Marie Vincent, Uy, Aberia, Erica,
Present po, ma'am. Present po.
Lorraine Abobo, Lorraine Abobo, Zoe Afable,
Present.
Alde Gail, Alde, Ambil,
Present.
Amoyo Raven, Amoyo, Ampong Joyce, Ampong,
po.
Apura Julia,
Present.
Apura,
Present.
Astakaan Nash, Olima,
Present, ma'am.
Dator Jan,
Present.
Dator, De Los Santos,
Present, ma'am.
Jolola,
Present po.
Lucena,
Present po.
Gapate,
Present.
Guevara, Lavado,
Present.
Marasigan,
Present.
Menzon,
Present po, ma'am.
Peralta,
Present, ma'am.
Promeda, Puno, Jaslyn, Puno, Sakmar,
kalak ng computer.
Sakmar,
Present po.
Suwal, Monroy, Angel, Monroy. Okay, so let's take section Santol. Say present once your name is called. Abusejo, Joshua, Anyosa, John Joseph, Are, Mark, Bulanadi, Catalo,
Present.
Delantar, Earl, Delatar,
Present po.
Labro, Andre,
Present.
Lily. Labro, are you there? Labro present. Labro. So Labro is not around. Lilia, Renz, Rosalejos, Samson, Traboco, John Ray. Okay, so for section Santol, of 11 boys who are enrolled in this subject, only 3 are present. Abelia, Adamas,
Present.
Okay, very good. Alaya, Francine,
Present po.
Amos,
Present po.
Hannah,
Present.
Apura, Denise,
Present.
Banario, Joselpe, Banario, Beato Andrea, Cepeda,
Present.
Cecista, Kiana,
Po. Present.
Doldolia, Jazare, Dulfo,
Present
Marian,
po. Present.
Factor Cristel, Factor, Pentes, Denisa, Pentes, Galupe, Gapud, Tricia, Garcia Angel, Gura Liana,
Present.
Hilaria Hazel,
Present.
Haboli Shira,
Present po.
Latoja,
Present.
Mendoza, Giselle, Mendoza, Montes, Padilla,
Present. Present po, ma'am.
Isaiah Pagayanan,
Present po.
Alison Puno, Yeza, Ramirez, Ninia,
Present, ma'am.
Rejang, Cherise, Sabido Grace, Sabido,
Present. Present, ma'am.
Turla Ella, Okay. So as promised, I will be giving, no, as promised. I will be giving actually an additional points for those who are attending my classes. Kasi napapansin ko, konti lang, especially for section Santol, konti lang yung, ma-attend ng lesson discussion, but many prefer to take the summative test rather than listening to the discussion. I just wonder where you get answers during the test. Okay, anyway, so may I ask you to turn on your camera? Okay, so we will be discussing today we'll be discussing today protein synthesis, which is actually still a part of module 4, lesson 2. Module 4, lesson 2. Okay. Now, protein synthesis is actually a very important process This is a very important activity that is going on 24/7 inside the cell. It is through this that the cell survives. Okay, now for a short recap on the discussion last time, so I was able to give you an overview of the central dogma of molecular genetics. Now, to give you again an overview of what we discussed last time, to give you an overview of the central dogma. So it starts actually from the DNA. Now, it is in the DNA where genetic information is permanently stored. So the genetic information is stored in the DNA and is copied by the RNA and translated into protein. Now, in protein synthesis, to give you an overview of this, now in protein synthesis, the language of DNA is translated into the language of protein. Please take this down. Again, in protein synthesis, the language of DNA is translated into the language of protein. Now, in protein synthesis, two major processes are involved, and these are transcription and translation, which we will discuss later. Okay. Now, replication is not a part of protein synthesis, but this is a part in the central dogma of molecular genetics. Now, replication again, this is what we discussed last time. Now, in replication, DNA makes an exact copy of itself. DNA makes DNA. Now, as what I've said just a while ago, the genetic information is stored in the DNA, which is copied by the RNA. Kasi nga, DNA can't go out of the nucleus, since DNA is permanently inside the nucleus. So how can this genetic information be copied, be translated into protein? So the RNA makes a vital role of copying the genetic message in the DNA, a process we call as transcription. Okay, so the transcribed genetic message will then be translated into protein through a process which we call as translation. So as what I have said just a while ago, protein synthesis involves two major processes, and these are transcription and translation. Okay, so are we all ready for the discussion of protein synthesis? Are we all ready?
Yes, ma'am.
Okay, thank you for that.
Yes po, ma'am.
Okay, thank you very much. Now, if in replication, the process involves a number of enzymes, again, in replication, a number of enzymes must be there for the process to be realized, for the process to become possible. Now, transcription and translation likewise involve some enzymes. Okay, so the focus for today's discussion is actually on how a protein is made inside the cell. If we go back to our first discussion in the cellular parts and the cellular components or the biomolecules present inside the cell or present in a cell, the cell is basically made up of protein. Only a few percentage is present in carbohydrates, nucleic acids, and fatty acids. Most of the cell is protein. So for this cell to go on living, it must make itself proteins. The question is, how can this protein be made inside the cell? So that is the focus for today's discussion, making a protein, a process we call as protein synthesis. Okay. Okay, so making a protein. DNA is divided up into functional units called genes, which may specify polypeptides or proteins and protein subunits. Now, when we say polypeptide, this is actually a short segment of a protein molecule. Protein is a macromolecule. Dako inihiya nga molecule. Now, a cell, it is difficult for a cell to make a whole protein. So what the cell do is it makes segments of a protein, which we call as polypeptides. Now these polypeptides are then joined together through amino acids. Through peptide bonds rather. So these polypeptides, which are basically made up of amino acids. So these polypeptides are joined together by peptide bonds to make a protein. Now in protein synthesis, as what I have said, in protein synthesis, what the cell makes is only a short segment of a protein or short segments of proteins. Again, the short segments of proteins, which we call as polypeptides, are joined together. They are joined together by means of peptide bonds. So pagdudurugtungon inhirang mga peptides, and it is only then that a protein, the whole molecule of a protein, is made. Let's say, for example, nakunta nga protein. Melanin, for example. Melanin is a protein that is responsible for the color, for the color of an organism, for the color of the eyes, the color of the skin, the color of the hair. So the protein responsible for this is melanin. Melanin is made up of so many amino acids. So itong protein, itong cell, di man hiya makakahimuhin bugos nga melanin. What it do is it makes polypeptides. Now these polypeptides are joined together for the cell to make the whole melanin. Okay, so I hope you were able to get it. So DNA is divided up into functional units called genes, which may specify polypeptides or functional RNAs. Now, aside from polypeptides, DNA also makes the 3 functional RNAs. And these are the messenger RNAs, the transfer RNAs, and ribosomal RNAs. Again, aside from proteins, DNA also make the functional RNAs. So it is the DNA that makes mRNAs, tRNAs, and rRNAs that are all involved to make a protein. Okay. Many genes provide instructions for building polypeptides. Polypeptide is just another word for a chain of amino acids. How exactly does DNA direct the construction of a polypeptide? A gene that encodes a polypeptide is expressed in 2 steps. In this process, information flows, again, as what I've said, from the DNA in order to make a polypeptide, or in order to make a protein. RNA must copy the genetic message in the DNA. And this copied genetic message is then translated into protein. Okay, so from DNA to RNA to protein. This is the directional relationship which we call as the central dogma of molecular biology or the central dogma of molecular genetics. Again, as what I've said just a while ago, this process involves two major steps, and these are transcription and translation. Okay, we go to transcription. Transcription from the word transcribe. Transcribe. So transcription is a noun. To transcribe is a verb. So what does it mean in your English? What do you mean by transcribe? To transcribe is transcript. Must go transcript of records. Transcript or transcription. Transcription is a noun from the word transcribe. To transcribe means to, anybody? To transcribe
Copy
means
something.
to, very good.
Make a written copy.
Okay, make a written copy or to copy. So transcription is simply copying the genetic message that is in the DNA. So that is what we mean by transcription. Again, di dahito DNA aada ang genetic information. Genetic information. Aada ang genetic message. Now, for this genetic message to be translated into a protein, for this genetic message to become a protein, it must be copied by an RNA molecule. Okay? So this step is called transcription because it involves rewriting or transcribing. Rewriting, kinokopya niya. Kinokopya. Ginsusurat, ginkukuha, ginkokopya niya or gintranscribe. The DNA sequence and a similar RNA alphabet requires the RNA. Okay. Copying of the genetic message is made possible by the messenger RNA. Again, it is the mRNA that copies the genetic message that is in the DNA. Masaring konta. I'll give an example. Ang message na saring konta, mahusay. Mahusay. So, itong mahusay actually involves a number of proteins. It involves a number of proteins. So, para maging mahusay itong isang organism, iton hiya nga message nga mahusay nga aad ha DNA kukopyahon hit mRNA. Para iton hiya nga message nga mahusay, mahimo hiya nga mga proteins, tapos when these proteins nga nagko-code hin mahusay, when these proteins are expressed, magawas itong organism na mahusay. Okay? So whatever is your color, whatever skin, whatever color of the skin you have, it has something to do with the message in your DNA. Okay? Which is translated into proteins. Again, the RNA molecule or the functional RNA molecule that involves in the copying of the genetic message that is involved in transcription is the messenger RNA. Messenger RNA. Okay, so we have it here. Okay. Now, to give you an overview of transcription, I will let you see a video clip or a short, just a short segment of the video clip that involves transcription. Ano to? Share screen. Okay, so let me repeat.
Excuse me, ma'am.
Yes.
Pareho po ba it sound?
Di ka muna batin sound.
Pareho.
May anyway, I'll just share this. I'll just upload this into Google Classroom. Okay. May ano ba tikit sound? Dihit akon. Wala. Anay ha? Oh. The green one there is actually the enzyme RNA polymerase. It is now synthesizing RNA. What's this? It is now synthesizing RNA nucleotides that it copied from the DNA. Now, the DNA here serves actually as the template in making this transcript. Now, from the nucleus, the RNA molecule, bringing along with it the message from the DNA, will now go to the cytoplasm, where the genetic message is translated into protein. Okay. Okay, so the whole process of transcription, since it is a process, basically it involves certain steps. So the first step in transcription is initiation. Okay, so initiation. Now, copying the message of the DNA doesn't happen just anywhere. It must have a specific site where copying of the message starts. And this site where the mRNA copies the genetic message is called as the promoter. It is called as the promoter. Okay, so we have it here, promoter. Okay, nababasa pa? Okay. So initiation is the beginning of transcription. It occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. Now the RNA polymerase, diba, normally DNA is always zipped. Kumbaga, the adenine is bind to a thymine through a hydrogen bond, and guanine bonds to a cytosine through a hydrogen bond also, or through hydrogen bonds. Okay, now, so generally speaking, the DNA is always closed. It only opens when it is replicated. It only opens when it is copied. Kuha. Again, DNA is always closed. It only unwinds, it only opens up, it only unwinds when it is replicated. So during replication, the DNA unwinds to make another copy of DNA. Now, another way by which DNA opens up, or where DNA unwinds, is during transcription. Now, it is the RNA polymerase that unwinds the DNA during transcription. Again, it is the enzyme polymerase that binds the DNA so that that DNA will unwind. When the DNA unwinds, it is only then that the genetic message in the DNA will be copied. Kuha, nga sa rin konta, ang message mahusay. Para makopya iton hiya nga gene nga mahusay, that DNA must unwind. So during transcription, it is the RNA polymerase that unwinds the DNA molecule to copy the genetic message. Now, that specific site or that specific region where the RNA polymerase binds to copy the genetic message is called as the promoter. Again, it is called as the promoter. Now, again, copying the genetic message doesn't happen just anywhere in the DNA. May adahiton niya a specific site where transcription starts. In the promoter, where transcription starts, has a 3-base sequence. TAC. TAC. Okay, maka-annotate na kita. DNA kontahiya. TAC. Supposing this is a segment of the DNA molecule where the RNA polymerase will copy. Now, again, as what I have said, the RNA polymerase will look for a region where it will start copying the message. Well, it will start copying the message. Okay, now. It will start in a region where when RNA synthesizes its nucleotides, it will be this one. R. Hiya. So, nagkokopya na hiya. Nagkokopya na hiya. Now, what I have said, the RNA polymerase will not start, it will not copy just anywhere. So, it will look for a site nga when it copies or when it will synthesize RNA nucleotides, it will become T will become A. What is the complementary base pair of adenine and RNA?
Thymine.
There is no thymine in RNA. There is no thymine in RNA.
Uracil.
Very good.
Uracil.
Uracil. What is the complementary base of cytosine?
Guanine.
Guanine. Very good.
Guanine.
Okay.
Guanine.
Okay. Now,
Guanine.
so must start itong copying itong message in a codon called methionine. So AUG. This is ito isang pagkokopya kasi itong RNA polymerase is by 3 base sequence. Again, 3 base sequence which we call as codon. 3 base sequence. AUG. So 1, 2, 3. 1, 2, 3. Adenine nucleotide, a uracil nucleotide, and guanine nucleotide. So ito ipagkokopya by threes or a sequence of 3 nucleotides or 3 base sequence which we call as codon. So itong pagkokopya and transcription, copying the message should start with the codon AUG. It will always start with AUG. Thus, in transcription, AUG is considered as the start codon. It is called as the start codon. Okay, Diritia must start it on ACC, TAA, because when RNA copies it, it will not become AUG. Okay, let's say, for example, anadida ATT TACT. Now, it will not start here. It will not start here. ATT, because when RNA will copy it, it will become ano na ba? Kita DNA. Okay. So when RNA will copy this one, ano't magiging nucleotides niya? T will become
A.
AAU.
A, very good. A
AU.
AAU. What about this? What about this? ACG. Okay. That was what I've said. That's what I've said just a while ago. Copying the genetic message will always start with a start codon. So it will always start with AUG. So from this, palihogla. Okay, thank you for that. Okay, now, in this segment of the DNA, where do you think will RNA starts copying the message? Is it in TAC, TGT? Okay, very good. In TAC. So magbibiling hiya in TAC so that when it will synthesize its nucleotides, it will become A, U, G, and will continue the message. Okay, so this is initiation. So initiation is the beginning of transcription. It occurs when the enzyme RNA polymerase binds the origin of a gene called the promoter. This signals the DNA to unwind so the enzyme can read the bases in one of the DNA strands. The enzyme is now ready to make a strand of mRNA with a complementary sequence of bases. Again, it will look for AUG. It will look for AUG. In other words, this site here, inihiya, this site here, inihiya, this site in the DNA is what we call as the promoter. This is now the promoter, or the initiation site, as we call it. So this is the initiation site with the initiation codon. Okay. Now, once RNA recognizes the start codon, copying of the message will now proceed. Copying of the message will now proceed. So from AUG, so ano naman it masunod? So TAC, okay. So the next set of nucleotides is GGC. So how will the RNA copies it? So the pagkopyan TAC is AUG. So from the language of DNA into the language of RNA. So AUG, and then it will copy the message in its own language. So it will become C, very good. C, G. Next, GGT will become
CCA.
A, and so on. Now, this step of transcription is what we call as elongation. Again, after recognizing the start codon, after recognizing the promoter or the site where transcription will start, RNA polymerase will now proceed copying the message in the DNA. And we call this step as elongation. Elongation. Okay. Copying of the message and elongation, copying of the message will go on. It will in this part, the RNA polymerase will keep on synthesizing RNA nucleotides that will complement the nucleotides in the DNA. Copying of the message will only stop. It will only stop. Kasi pag may beginning, definitely there will be an end. Kung may ada start codon, definitely opposite to that, may ada niya May ada niya. Ano't opposite? Hitong start codon.
End codon.
Stop codon. Ano? Bali, kung sentence hiya, kung sentence nga nagtikang hiya, let's say for example, ang sentence na sa ring. The boy was okay, kompleto hiya. So, may ada beginning. Let's say, for example, the here is the start codon. The boy was sad. So, to end the sentence, how will you end the sentence? What punctuation mark will you use?
Period.
Very
Period.
good. Period. So period na nga ni, meaning wari not masunod. Ano? Okay, wari not masunod na word. Kay period naman dida. Anyway, just like in copying the message. So the RNA polymerase started it with the start codon AUG. After recognizing AUG, RNA polymerase will now keep on copying the message in the DNA and will only stop once it will recognize a stop codon. Adalat hiya maudong. So makatara na nga riyan stop codon, meaning period na hiya. Okay. Now we call that step as termination. Okay. Okay, now there are but 3 stop codons that are identified. So there are 3 stop codons: UAA, UAG, or UGA. Ano? Let's take for example, ang nasunod dinhi is, ay nata dinhi konta, ang nasunod is A, T, T, A, T, T, ATT. Okay, so how will the RNA polymerase copies it? How will it copy?
UAA.
A. Okay. Do you think the RNA, once it was able to identify UAA, do you think the RNA polymerase will go on copying the genetic message in the DNA?
No.
No.
No, because UAA is
A stop codon.
The
stop codon. Very good.
Codon.
Signaling the RNA polymerase to stop copying the message. Okay, so UAA actually does not code for any amino acid. Okay, it doesn't code for any amino acid. So ito magiging resulta, RNA polymerase will stop copying the message, a step called termination. So copying of the message will now end. The mRNA, the messenger RNA will now detach from the DNA molecule. It will now detach from the DNA molecule. Okay? Okay na? Okay. Hala? Wala magandang sound. Magandang sound. Magandang sound nga. Anyway. Okay. So, that is transcription. Now, we go to the next one. Hala, basa talaan na yun. The location of transcription is also different between prokaryotes and eukaryotes. Eukaryotic transcription takes place in the nucleus where the DNA is stored. Again, this is very important. Transcription happens inside the nucleus. Transcription happens inside the nucleus in eukaryotic cells. Itong pagkokopya adhan nucleus. Why? Because DNA is in the nucleus. Okay. In prokaryotic transcription, since it doesn't have a nucleus, transcription takes place in the cytosol of the cytoplasm. So, transcription in prokaryotic cells happens in the cytosol or in the cytosol of the cytoplasm. Okay, what man niya nucleus? Okay, we go to translation this time. Translation. So nagkopya na. Ginkopya na ang message. Ginkopya na ang message nga mahusay. So dire ito niya magiging mahusay if it will not be translated. Okay? So in translation, the sequence of the mRNA is decoded to specify the amino acid sequence of a polypeptide. The name translation reflects that the nucleotide sequence of the mRNA sequence must be translated into the completely different language of amino acids. Now, again in the Central Dogma, the language of DNA. The language of DNA is translated into the language of protein. The language of DNA. What is the language of DNA in the first place? Liana. Liana, are you there? Liana. So what is the language of the DNA? What do you think? Any volunteer? Ano itong language itong DNA? I'll give 3 points to the summative test. So anybody who will answer, who can answer the question. So what is the, please identify. Identify your name.
The genes?
Genes, another.
ATGC.
For nucleotides.
Anna? Who answered that? Who answered nucleotides?
And
Who answered nucleotides?
for nucleotides.
Who answered nucleotides? Who answered nucleotides? May binatik nucleotides. Who answered nucleotides? Please identify. Who are you?
Debbie po.
Debbie, thank you for that. So Debbie plus, pero naksiring Debbie? 3 points?
3.
Okay, record ko diretso. Menzon. So plus 3. Okay, now the language of DNA is, thank you for that, Debbie. The language of DNA is nucleotide. Now this nucleotide will then be translated into the language of protein, which is amino acid. So from nucleotide, it will be translated into the language of protein. So from nucleotides to amino
Amino acid.
from nucleotides to amino acids. So this is now what we call as transcription. Ang pagkopya, di ba, ginkopyahan RNA. Pagkopya itong RNA, it is still nucleotides. It is still nucleotides. Kasi itong ginkakaibala dida, thymine is replaced with uracil, but those are still nucleotides. So the language of nucleotides in the DNA, which is copied by the RNA, is translated into the language of proteins, into the language of protein called amino acids. Okay? Anay ha, kami ada na sulod. Oh, wait lang. Meron ba? Okay. Okay, so, klaro na? Now, in translation, it involves these three structures: mRNA, the tRNA, and the ribosomes. Again, for translation to occur, it must have the mRNA. So itong mRNA din must have or must carry the genetic message that it copied from the DNA. So may adanat niya, we call this as the transcript. The transcript. Okay, the messenger RNA or the transcript molecule. The mRNA with the message from the DNA. The tRNA and the ribosomes. So itong tRNA, drawing ako kita ha. In tRNA, this part here is the anti-codon. So may adahit dida, this part here. It's called as the Okay, while this part here, this is the site for the codon, the anticodon, while this is the site for the amino. Part here, dinhi adat amino acid, while dinhi ada it anticodon. Dinhi mRNA aanhi it codons. Codon. Dinhi adat dida itong codons. Dinhi naman na tRNA is where we can find the anticodon. This part here is for the anticodon, and this part here, or this site here of the tRNA is where we can find the amino acid. Now, ribosome. The ribosome, a ribosome has two parts. So this is the small part and the small amino acid and the large amino acid, or large amino acid. The small ribosome, the small unit of a ribosome and the large unit of the ribosome. So this is the small unit. Small unit of the ribosome, while this one is the large unit of the ribosome. In the small unit are three sites. And not three sites. So this is E site. This is the P site, while this one is the A site. Okay, so again, in the small unit of a ribosome, it has 3 sites. E for exit, site. P for peptidyl site, and A for
Accept.
amino acid or acceptor. So E for exit site, P for peptidyl site, and A for amino acid or acceptor site. Okay? The 3 sites in the large or small unit, class?
Small unit.
Large.
Small unit.
Small unit.
Small unit. Okay? Small unit, ha? Okay. Now, ginkopya na niyo? Can I erase this one now? Nakopya na? Okay, sige. Okay na. I-erase ko na, ha? Okay. Okay, now. I will let you see or watch a video clip again on translation. Nay, wait lang. Okay. Can you see? Can you see, class?
Yes, po.
Yes, ma'am.
Sana may other sound. Sana may other sound.
Yes.
Can you hear?
No.
No.
No, ma'am.
Direct.
Po, ma'am. Opo, ma'am.
Yes, ma'am.
No po.
Okay. So, repeat kita. Okay. Okay. So this one here is the transcript. This is the messenger RNA that carries the message that it copied from the DNA as in here. Okay. So inihiya nga mga codons, AUG, CCG, UA, C, and so on. So these are the nucleotides that it copied from the DNA. So aanhidinhi ang genetic message. For this message to be translated, it must, the mRNA must seek the help of a tRNA. So itong tRNA, may ada ito niya what we call as the the anticodons. The anticodons, itong anticodons, may ada ito niya nucleotides that will complement with the nucleotides in the mRNA. Remember that this is a tRNA. And this one is an mRNA. So, U complements with A, right? Are you still there, class?
Yes.
Yes po.
Yes po.
A
Yes.
complements with U, C with G.
Yes, ma'am.
So, waray ibang nga nucleotide nga ma-combine itong AUG but AUAC. The tRNA UAC. Now, didi naman hiton iya opposite end. This site here carries now the amino acid. So, AUG actually codes for methionine. The F here means formyl methionine, which simply means nga sa mering nga ni formyl methionine, meaning start codon iton hiya. Start codon. Ha? Kaya kung diri hiya start codon, AUG la nga ni, iton aanhidinhi is simply methionine. Okay? Okay, let's continue. Okay, now. The small and the large units of a ribosome are always separated. Bulag it hira. Magko-combine la iton hira if there is a message to be translated. Magko-combine la iton hira if there is a messenger RNA carrying a genetic message that is to be translated. Waray nga ni i-translate or waray nga ni mRNA na aada ha cytoplasm, then these two will separate. Okay? Next. Okay, now, the AUG, the start codon, di ba tutulong nga sites? Exit, di ba tulong nga site ano? Exit, E site, P site, and A site. Exit, peptidyl, and acceptor site. Itong AUG, which is the start codon, will go directly into the peptidyl site. Please take note of this. Diretso hiya maka diha peptidyl site. While the next codon will stay in the acceptor site. Nakuha? Were you able to get it? Again?
Yes.
Again, the start codon will go directly into the peptidyl site. Ang nasunod ha iya nga codon, CCG, will stay in the acceptor site. So, pagkadahan AUG ha peptidyl site, it is only then nga maabot an anticodon, an transfer RNA with UAC anticodon with the methionine amino acid. Okay? So, igtra-translate na hiya. Igtra-translate na ang AUG into methionine. So, ang CCG, waiting na hiya. Waiting. It is already in the acceptor site. So, waiting na hiya. Okay? Okay. Diba? Na-translate na ang AUG into methionine. After which, diba, nag-join na ang tRNA with CCG. CCG codes for proline. After translating AUG into methionine, this AUG will now proceed to the exit site. Okay, it will now advance into the exit site. Tapos, marirelease na hiya. Okay, so another tRNA will move into the A site. Okay, look at that. So UAC will now be translated into tyrosine, and this one here will now go to the exit site and will detach. Okay, it will be detached from the mRNA. Now look at this, No, okay, now. As what I've said just a while ago, a stop codon doesn't code for any amino acid. It doesn't code for any amino acid. So, waray ha iya makada nga tRNA because there is no tRNA that will complement, there is no anticodon that will complement with UAA. Okay? So, translation will stop until here. So, ini naman hiya nga mga amino acids, makakapurma na hiya in what we call as a polypeptide. So, nakahimo na hiyahin short protein molecule. A short segment of a protein molecule. Okay, balik. So we call that actually as a polypeptide, which is basically made up of amino acids. Okay, so we'll go back to the module. Okay. Now, questions on translation? Question. Feel free to ask. Children, are you still there? Ada pa ka mo?
Yes po, ma'am.
Yes,
Yes, ma'am.
So feel free to ask.
Yes,
Ada
ma'am.
pa, okay, Ana?
Yes, ma'am.
Now, during translation, we go to the genetic code. This is very important. During translation, the nucleotide sequence of an mRNA is translated into the amino acid sequence of a polypeptide. Specifically, the nucleotides of the mRNA are read in triplets. So meron nga ni triplets, a group of 3 or 3 base sequence. Masaring konta. A, G, C. So 3 base sequence, and we call this as a codon, a triplet or a codon. So there are 61 codons that specify amino acids. Each codon specifies 1 amino acid. 1 codon is a start codon that indicates where to start translation, and this is AUG. The start codon specifies the amino acid methionine. The start codon AUG actually specifies the amino acid methionine. So most polypeptides begin with this amino acid. 3 other stop codons signal the end of the polypeptide. These relationships between codons and amino acids are called the genetic code. Now, here is a genetic code chart. And this is how you will use this. This is very important, and I want you to know how to use this because you will be using this during the summative test. Let's take, for example, the codon is AUG. How will you know that AUG codes for methionine? By using this chart. So this is the first letter of the codon, the second and the third. First, second, third. So first, so look for A. So ada here A, A. So A. U in the second letter. Anhi ka din himapili. First letter, anhi ka din himapili. So A, U. Itong third letter naman, kung hain ani mo first, so aanhi ka din himapili. So A, U, G. Combined, so AUG codes for methionine. Okay, it codes for methionine. Another example. C, the codon, C, G, U. So the first letter is C. So adikadide is C. The next letter is G. So adikadide. The second letter is G. So din hiya C. The third letter is U. So adikadide mapili. U. Pagtarampuha. C, G, U. And CGU codes for arginine. Okay, one last example. U, A, A. So what does it mean? The first letter is U.
Stop.
A and A, meaning U, A is a stop codon. Stop, meaning no other what's this? Translation will stop because there is no amino acid that will code for this. Okay, so meaning stop na, period na, and it was able to make one polypeptide. Kuha? Okay, so okay, let's have a drill. I'll give you 3 exercises. Hala, sige daw. G, A. What does it code for? What does it code for?
Glycine.
Okay, gly or glycine. Okay, next. U, G, U. UGU. Okay, cis or cysteine. Cis or cysteine. Last. Or threonine. Okay. Add it on here. Okay. PHA phenylalanine, LAU lysine, ILE isoleucine, methionine, valine, serine, proline, THR threonine, alanine, tyrosine, histidine, glutamine, asparagine, LY is for lysine, ASP for aspartic acid. We have it here. AS. Okay, so GAU, GAC are aspartic acid, amino acids. So we have glutamic acid, cysteine, tryptophan, arginine, serine, arginine, and glycine. Okay, now, aside from this, there is another codon or genetic code chart, which is easier to use than the previous one. So this is how you will use it. How you will use this one. Let's take, for example, GGA. GGA. So this is the first letter. The biggest is the first letter. Second biggest is the second letter, and the smallest is the third letter. So GGA. So this is how you will use it. GGA, glycine. UGU, UGU, cysteine. ACG, ACG, threonine. AUG, AUG, methionine. CGU, CGU, arginine. And UAA, UAA, which means stop. Okay, hain mas masayon, class? The previous one or this chart?
The latest one. One po.
This one. So this is easier than the previous one. Pero in most biology books, the genetic code chart is this. So gintot doon tala ka mo, kaya you might enroll in medical course, tapos ditohan iyo libro, as yan nasugad hini, So at least you know how to use it. Okay, kasi merong genetics sa medical school or sa medical course. Okay. Okay na? Okay na?
Yes.
Yes po.
Okay. So,
Yes po.
codons. So cells decode for mRNA by reading their nucleotides in groups of 3 called codons. And here are some features of codons. As in mga characteristics of codons. Most codons specify an amino acid. Okay. 3 stop codons mark the end of protein. Please take note of this. 3 stop codons mark the end of a protein. 1 start codon, AUG, marks the beginning of a protein and also encodes the amino acid methionine. Okay? Codons in an mRNA are read during translation, beginning with a start codon and continuing until a stop codon is reached. mRNA codons are read from 5' to 3', and they specify the order of amino acids in a protein from N-terminus to C-terminus. Diba? Sagad kasi ito niya. Sugad. So, inidinhi, let's say for example, 5' to 3'. So 5' to 3'. This is from N-terminus. So DD, the N-terminus. This is the N-terminus. While this is the 3. The C-terminus. Okay, so this is the C-terminus. Ano tada N-terminus? Ano tada C-terminus? Let's move on. What does 5' and 3'? 5 refers to the orientation of nucleotides of single strand of DNA. And the 5' and the phosphate group of the first nucleotide in the chain. And we all know this. The phosphate group is attached to the 5th carbon, the sugar ring, which is why this is called as the 5' end. At the other end, called the 3' end, the hydroxyl of the last nucleotide added to the chain is exposed. The hydroxyl group is attached to the 3' carbon and sugar ring, which is why this is called the 3' end. Okay, now, a peptide has 2 ends. The end with the free amino group is called the N-terminus. So this is the end with a free amino group. While the end with free carboxyl group is the C-terminus. Ano taanhi? The hydroxyl group. Peptides are named from the N-terminal acid residue to the C-terminal amino acid. So this is the amino acid structure. This is the amino group. So this is the N-terminus, while this one is the C-terminus.
Bye, Mom.
Okay. Are you still there?
Yes po.
Yes.
Yes, Ma'am.
po.
Okay.
Yes, ma'am.
Pinabatikhin, goodbye, ma'am. Okay, so translation is the process by which a protein is synthesized from the enzyme. Just to read it, anyway, you have a copy of this. I think I have uploaded this in the Google Classroom many weeks ago. Do you have this?
Module 2. Lesson 2.
Yes, yes. Pero ini, gin-add ko na lang ini yung module. Gin-add ko. Okay. So this is the elongation of translation. Now, in translation, it also follows certain steps. First is initiation, where
Initiation,
AUG,
elongation, termination.
yes, where AUG stays at the peptidyl. Initiation yan, where the AUG stays at the peptidyl site of the small unit of the ribosome. Tapos makada na haiya ang tRNA carrying the methionine amino acid. So after translating the first codon, translation will proceed, and that step is now elongation. Translation will be terminated when any of the stop codons, UAA, UAG, and UGA arrives in the ribosome. Since there is no amino acid that will code any of this, UAA, UAG, and UGA, then translation will be terminated. It will end. Okay? So it signals these three stop codons signal the ribosome to stop synthesizing the protein. Okay, so feel free to ask before we have a short exercise. Okay, now we'll start from replication. So use the DNA code provided and fill in the complementary DNA strand. TAA. So what is the complementary
ATT.
ATT. Very good. Next.
GCT.
GCT. Next. AGC. Very good. What about the other one? GTC. Next. ACT. Very good. Next. GAA. Which nitrogen base can't you use during replication?
Uracil.
Uracil, very good. Thank you. Uracil. Since uracil is present only in RNA. Kasi replication, DNA yan. Walang uracil sa replication kasi DNA yan. Next, what enzyme is involved in unzipping of a segment of DNA?
Helicase.
Helicase, very good. Thank you. So, do you still remember Okazaki fragments?
Yes, ma'am.
Where can we find Okazaki fragments? Leading or lagging
The
strand?
ones with the
Where can we find Okazaki fragments? Leading or lagging?
lagging strand.
Lagging, very good.
Lagging strand.
So how will you describe Okazaki fragments?
Little gaps in the lagging strand.
They are found in the lagging strand. Okay, very good. They are short segments of a DNA molecule. Okay, so we go to transcription. Use the DNA code provided to copy an mRNA message. So this is DNA. So what will be the mRNA for this? TGC.
ACG.
TGC.
ACG.
ACG. Next.
ACU.
ACU. Very good. Next. GG. Very good. GGA. Thank you. Next. Letter B. CGC. Next.
GAU.
GAU.
GAU. Okay, GAU. How many types of RNA exist? 1, 2, 3, 4, 5.
3.
3. Name each type. What are the 3 types?
mRNA,
Messenger RNA.
tRNA, rRNA.
RNA.
Okay, very good. mRNA, tRNA, and rRNA. What is the role of mRNA in protein synthesis?
It copies the
Information.
It copies.
A genetic message.
the genetic.
Okay, very good.
Genetic
The
information.
genetic message in the DNA. Okay, so we go to translation. Use the genetic code table to determine the amino acid that corresponds to the mRNA code given. Okay, CCA. Use the genetic code chart. Do you have it there?
Proline.
Lean.
Proline. UGU.
UGU. Cysteine.
Okay, cysteine. It is pronounced as cysteine. Cysteine, hadera cysteine. Cysteine. AAU.
AAU. Asparagine.
Asparagine.
Asparagine. AAU. Asparagine. Asparagine.
Yes.
Asparagine.
Asparagine.
Nay, kita unta. Okay. AAU, wala. Kita unta. Asparagine, asya ba?
Asparagine.
AAU. Asparagine.
Spargin.
Okay, asparagine. So next. GGG.
Glycine.
Glycine.
Glycine, very good. ACA.
Threonine.
Threonine. Okay. Now, what about if during the summative test, I will give you this one, masaring konta. Masaring konta. We'll make a table first. So, table. DNA, mRNA, tRNA. So this is DNA. So, how will you answer it? Now, DNA is CGT. So you will supply mRNA, tRNA, and amino acid. In number 2, the given is UGA. So you will supply DNA, mRNA, and amino acid. Number 3 is, the given is GUC mRNA. So you will supply DNA, tRNA, and amino acid. Okay? So let's take number 1. CGT. So what will be the mRNA?
GCA.
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 ECU.
GCA.
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 Ano?
GCA.
G,
GCA.
C, A. Siya? Okay, next. What will be the tRNA?
CGU.
What will be your basis? DNA or mRNA?
Mrna.
mRNA, very good.
RNA.
So what will be the tRNA? What will be the anticodon?
CGU.
In mRNA is the codon. In tRNA is the anticodon. So what will be the anticodon for GCA?
CGU.
There is no thymine in RNA. Remember, this is
CGU.
U. Very good. So what will be the amino acid?
Arginine.
What will be your basis? Is it tRNA or mRNA?
tRNA.
TRNA.
Ano itong gintra-translate? mRNA or tRNA? Hide itong message in the mRNA or in the
RNA.
Ano RNA? Kinudot din na M and T. So translate mo into amino acid. The message in the mRNA. Hide itong message in the first place. In the mRNA or in the tRNA?
mRNA.
Very good. Itong message ada mRNA kay ano? It is the mRNA that copied the message in the DNA. Meaning, itong i-translate din into amino acid will be the mRNA. Okay? So, what is the amino acid then?
Alanine.
Alanine. Does it code for GCA? Is alanine code?
Yes.
Okay.
Yes.
So, the answer here is alanine. Okay na? Okay na? Okay. Now, number 2 is
Yes po.
UGA. tRNA. So, what? Ano't imo primero nga bubuhaton? Will you supply the DNA first or the mRNA or the amino acid? Ano't imo primero? mRNA. Very good.
mRNA.
Here. So what will be? What will be the mRNA?
ACU.
ACU.
A, C, U. Next thing you will do is to
DNA.
supply DNA. Supply DNA. So what will be the DNA?
TGA.
What will be your basis? What will be your basis? What will be your basis? RNA or tRNA?
mRNA.
mRNA. Kaya hiya ang nagkopya, di ba? Hiya ang nagkopya. So mRNA, ito yung basis. So the answer will be
TGA.
G. Okay. Now we go to amino acid. So what will be the amino acid here? Threonine. What was your basis? mRNA again. mRNA. So pero itong basis, mRNA. Okay. So what is the amino acid for ACU?
Threonine.
Ha? Threonine? Okay. So GUC. What will be the DNA for GUC? GUC. What will be the DNA?
CAG.
What will be the tRNA for this? CAG. So CAG. Okay. So next, what will be the amino acid?
Glutamine.
Did you base tRNA or mRNA?
mRNA.
RNA, very good. So GUC codes for
Glutamine.
glutamine. Now you are learning. That is how the cell makes a protein. Okay?
Ma'am, ano yung amino acid?
mRNA yan,
Valine na
GUC.
Amino acid.
Valine.
Paano mo na yan?
Alin po, ma'am.
GUC. Oh, valine.
Valine.
Okay, valine. Okay, thank you. So, asan dapat itong iyong mga answer? Okay? Did you find it hard? Okay. Now try practicing at home. Try practicing at home. Where in the cell does translation take place?
Nuclei.
Translation.
Translation.
The ribosomes. Ribosome.
Where in the cell does this process take place? Diin, nga parte itong cell. In the cytoplasm, very good. Cytosol in the cytoplasm. Transcription occurs in the nucleus, while translation occurs in the cytosol of the cytoplasm. On which type of RNA is the codon located? Codon. Where is it located?
tRNA.
Oh.
tRNA.
Which RNA is the anticodon located?
mRNA.
Okay. Let's take the first one. On which type of RNA is the codon located? In
mRNA.
RNA. Okay. So mRNA. Which type of RNA is the anticodon located?
tRNA.
tRNA.
RNA. Very good. tRNA. Okay. So what is the another name for a chain of amino acids?
Polypeptide.
Peptide, very good. So what is the purpose of tRNA? What is the purpose of tRNA?
To translate the mRNA
mRNA.
to translate the mRNA
Translate.
to amino
T.
acid.
T means transfer. But transfer RNA. So what is the purpose of transfer RNA?
To translate.
To transfer.
Transfer, yes, to transfer.
To bring amino
acid to ribosomes.
Transfer the amino
Very good. To transfer or bring amino acid into the ribosome. So that is the function of the tRNA. Okay. Okay, meron pa ba? Okay. So, I'm glad you were learning or you learned something. So, for your assignment, you will do activity 3 in apply. Do activity 3 in apply. Yun lang. And then answer the assess. Write your answers or send your answers through PM. Okay na? So that will be your assignment. Activity 3 of apply and then the assessment. Okay na? Now,
Ma'am, sana po pag-pass.
anytime this week. Anytime this week.
Okay po.
PDF.
PDS.
PDF.
PDF. I think I have uploaded this in the Google Classroom. Meron po. Yeah, you find the in the Google Classroom. You find the file in the Google Classroom.
Ma'am, di ko po makita.
Google Classroom?
Yes po.
May Central Dogma.
Central Dogma. Sige po.
Oo, Central Dogma. Okay, so did you learn something today?
Yes po.
Yes po.
Yes,
Okay,
ma'am.
thank you. I will again check the attendance. So those who lasted until the end of the discussion will be the ones to be given additional points in the summative test. Okay? Say present once your name is called. We'll start from section Santol. Abosejo, Joshua, Anyosa.
Present.
Anyosa, present?
Present po.
Okay, thank you. Are, John Edward, Bulanadi, Mart, Catalo, Earl.
Present.
Okay, thank you. Delantar, Earl.
Present.
Okay. Labro, Lilia, Renz, Rosalejos, Samson, Raidel, Samson, Traboco, John Ray, Abelia, Adamas, Kera, Adamas, Alea Francine,
Present po.
Alea, Amos, Hannah,
Present.
Apura, Denise,
Present.
Banario, Josel, Banario, Beato, Andrea, Joy, Beato, Andrea, Beato is not around. Cepeda, Miska, Cepeda, Marina, Cepeda. Sisista, Kiana,
Present po. Present.
Doldolea, Jazzy, Dulfo, Marian, Dulfo, Marina, Marian. Factor, Fuentes, Denisa, Galupe, Hyacinth, Gapud, Tricia, Garcia, Angel, Gura, Liana,
Present.
Gura, Hilaria, Hazel,
Present.
Habuli, Shira,
Present po.
Latoha, Charnet,
Present.
Latoha, Latoha, Mendoza, Giselle, Mendoza, Montes, Jahara,
Present po.
Padilla, Jazel, Padilla, wala na. Pagayanan, Isaiah,
Present po.
Pagayanan, Pan, Allison, Puno, Yesa, Ramirez, Ninia, Ramirez, Ridyang Cheris, Sabido, Grace,
Present.
Urla, Ella. Okay. Padilla, wala na. Dulfo, Marian, wala na rin. Cepeda, Miska. Okay, so let's take section Mango. Apita,
Present. Present.
Raba, Mark,
Present.
Banaldia,
Present.
Bantiles, Bihar, Estrobo, wala na. Go, Hardillo, Ernst, Hardillo, wala na rin. Malinaw, Marasigan, Christian, Marasigan, Nicart,
Present.
Present.
Ogala, Jade, Panis, Seso, Solayaw,
Present.
Uy, Marie Vincent, Uy, wala na rin. Aberia,
Present po, ma'am.
Abobo, Lorraine, Afable,
Present.
Alde Gale, Ambil, Ana,
Present.
Moyo Raven. Ampong Joyce. Ampong. Wala na. Apura Julia.
Present.
Okay, so Apura Julia.
Opo.
Present. Okay, sige. Astakaan Nash. Nash. Colima Angeline.
Present.
Dator John Antoinette.
Present.
De Los Santos Krishna. De Los Santos. Wala na. Jolola Jocelyn.
Present po.
Docena Aliana.
Present po.
Gapata Jillian. Gapate. Wala na rin. Guevara Julia.
Present po.
Lavado.
Present.
Marasigan.
Present po.
Menzon.
Present po.
Peralta.
Present, ma'am.
Promeda. Francesca. Uno Jocelyn. Sakmar Riz.
Present.
Suwal. Monroy Angel. Okay.
Morning.
Last call for Monroy, present. So last call for Gapate, Jillian. Are you there? De Los Santos. Astakaan. Ampong. Alde Gale. Uy, Marie Vincent. Hardillo Ernst. Estrobo, Lance. Okay, so Anwaray ilog nga daan last part waray plus points. Anyway, so that ends module 4. And if the weather permits, then we will schedule the summative test this week. Okay, coverage will be the whole module 4. Whole module 4. So lesson 1 and lesson 2. If the weather will not permit, so if the weather becomes more aggressive, kasi merong parating na bagyo, we'll just have it in January. I hope you still remember the concepts, you still remember the lessons in January or by January or in January. Kasi matagal na yun. Siya nga, I would like to have it this week para presko pa. Okay, so that ends our lesson. Thank you for participating. I hope you learned something today. Goodbye, class.
Goodbye, ma'am.
Bye po.
Bye, ma'am. Thank you.
Bye po.
Bye-bye.
Thank you, ma'am.
Thank you. Bye-bye.
Bye, ma'am.
Bye, ma'am.